initiative taken by LJ Institute of Pharmacy. This is Joshi from Department of Pharmaceutical Technology. In today's video, we'll be learning about complexation and protein binding, part 2. But before moving further, please do like, share and subscribe our YouTube channel Pharma Ignite. Now, we have seen complexation, its definition and its application in pharmacy. So, in today's video, we'll be learning about classification of complexation. There are three types of subtypes of complexation, that is metal complexes, organic molecular complexes and inclusion complexes. Now, in every types of classification, there are subtitles which are involved. First one is metal complex. In this metal complex, metal is understood as central atom, and next one is the base, which is taken as electron donor group. Now, when the electron is donated to the metal ion, central metal ion, what will happen? There will be formation of complex, and this is formed by coordinate bond. Next, in this metal complex, first one is inorganic complex. So firstly, we need to understand Wegener's postulate. So first one is there are two types of valencies, primary and secondary. So primary is understood as ionic, and secondary valency can be understood as coordinate one. As you can see in the slide, there is an example which is given. So in the second one, or the second postulate of Wegener is same type of anion, radical and molecule may be held by one or both types of valencies, either primary or you can say secondary. Now third one is the central atom has a fixed number of non-ionic valency, or you can say coordinate number. Fourth one, the coordinate atoms occupy first sphere, or you can say coordinate sphere, and the other atoms will be having the secondary sphere, or you can say ionizable sphere. Now fifth one is the neutral molecule or ion may satisfy non-ionic valencies. In the sixth postulate, what Wegener has to say is the non-ionic valencies are directly specific, or you can say have directly specific position in the space. Moving further, the second subtitle is chelates. Now what are chelates? These are group of metal ion complexes in which substrate or ligand provide two or more donor groups to combine with the metal ion. So it can be ligand, can be didentate, tridentate or polydentate, and accordingly the name is given. Now this is also known as sesquiterring agent. Now what are they? They are stable water soluble metal chelates. Example is chlorophyll, or you can say hemoglobin. The next one in this is C and D, that is olefin and aromatic type. It is not much in use in basic pharmacy, but you can say to manufacture bulk drugs, intermediate drug analysis, you can use this. It involves Lewis acid-base reaction. While in case of complex can be separated from the solution and cannot be separated from the solution, and molecular compound can be separated from the solution. Next is drug and caffeine complex. The acidic drug can easily form a complex with caffeine. Example is benzocaine and procaine. Now, the mechanism which is involved in this is dipole-dipole force or hydrogen bonding between acidic drug and caffeine carboxyl group. Now, the second mechanism is interaction of non-polar parts of it. So, what is the application of drug and caffeine complex in pharmacy? First is to increase or extend or improve absorption and bioavailability of the drug, to inhibit or to increase or enhance the solubility and the dissolution rate of the drug. And third one is to taste mask. For example, you can make a chewable tablet of a bitter drug out of this complex. Second one is polymer complexes. The polymer complex needs to be understood well to understand the incompatibility which arises when the complex forms and it interacts with the containers and even the drug. So when they interact with each other, they generate incompatibility. For example, when the container and the complexes interact with each other, there will be certain you can say incompatibility is arise and the drug loses its action. Now, similarly, when the preservatives are present, these complexes also interact with these preservatives and they may lose or decrease the activity of the preservatives itself. So next one is picric acid complex. This picric acid is strong acid, but when it comes combined with a strong base, it will generate salt. But when this picric acid, being a strong acid, interacts with a weak base, it forms complexes. The best example is butacin. Butacin picrate, when interact with each other, this picric acid is antiseptic while butacin is anesthetic. So it can be used in burns and abrasions. But the major disadvantage of this picric acid is it combines with the carcinogenic agent and forms complex, which increases the carcinogenicity of this carcinogenic agent. So this is the major disadvantage of it. Next one is quinine-hydrogen complex. In this complex, there is overlapping of you can say pi electron molecules and hydrogen bonding for stabilizing the complex. The complex is between hydroquinone and benzoquinone, or you can say alcoholic solution of equimolar quantities of the both. They form a green crystal, and it is the application is it acts as an electrode to determine the pH. The last one is inclusion complex or occlusion complex. The word itself tells inclusion, to include something. It will trap the molecule in a cage-like structure. Okay, and the interaction are due to suitable molecular structure. The prediction of complex is a little bit difficult over here, but as such, there is a relation of host and guest molecules. So first one in this is channel type. Now channel means what? There is a host and a guest which is arriving. There is the guest molecules are unbranched molecules. The unbranched molecules will come and settle inside. So outer portion of this is hydrophilic in nature, but the inner portion is hydrophobic in nature. So those drugs which are hydrophobic can easily be included in this channel type. Now, what is the application in pharmacy? First of all, separation of isomers, or you can say you can analyze large molecules used in dermatological preparation like creams, lotions, and which will be interfering in analysis. So you can separate them very easily. Now next one is layer type. This layer type is there is again a host and a guest molecule, in which the guest is alcohol, glycols, hydrocarbons, and there in the host there is a channel. First is host, then guest, host and guest. This kind of arrangement is known as layer type of arrangement, and it is used basically because of its large surface area and used as a catalyst. Now the last one is the catheter. It is again a cage-like structure. For example, during crystallization, some of the compound of the host form a cage structure, and in this cage structure, this guest molecule gets entrapped. But the molecule should not be large enough that it cannot enter and small enough so that it can escape. It should be well fitted inside the whole molecule and settle inside. Again, the basic application is for separation or to store the volatile material like gases, and even to store the toxic substances. Next is monomolecular inclusion complex. In this monomolecular inclusion complex, as I have explained, the best example is cyclodextrin. Now, the outer portion of it is hydrophilic in nature. Inside it is hydrophobic in nature. The diameter vary accordingly. But here, cyclodextrin beta, okay, that is widely used in pharmacy. There are three types: alpha, beta and gamma. As you can see in the slide, different diameters has been written, and the suitable one is beta. Okay, so the guest molecule can be hydrophobic in nature, which can be protected from the outer environment. So now we have to analyze if complex is formed or not. For this, you require different methods to analyze the complex. So first method is continuous variation method. Second one is distribution method. For distribution method, it is similar to the separation method as we use in you can say separating funnel. Third one is solubility method. Last one is pH titration method, in which one is standard and one is reference. The standard one is the test sample. The test sample is a complex one, and you can say the reference one is which is not forming a complex. So last one, from here you can understand the method of analysis, different types of complexing agents, and even the classification of it. So